We come to the last talk, which is from Scott Megill from Coriell Life Sciences, Outcomes from CDSS-Guided Pharmacogenomics Implementation. Thank you.
So I have the enviable last slot of the day, which is great. It means I get to beat some horses that are already long dead. So just a little bit about us, if you don't know Coriell. So Coriell was founded all the way back in 1953 by Lou Coriell, probably most famous for working with Jonas Salk and commercializing and scaling up the polio vaccine. Through obviously a long period of research and building out biobanks and things like that, a research study was started in 2007 focused on personalized medicine. That spawned Coriell Life Sciences, which is a for-profit endeavor about 10 years ago, January of 2013. So I'm Scott Megill, President and CEO of Coriell Life Sciences. I founded the company as a spin-out from Coriell, from the Coriell Institute for Medical Research. So over lunch, it was sort of fortuitous. One of the individuals we were having lunch with said, why isn't everybody doing this today? Right? We're all here to say this is the right thing to be working on and the right thing to be doing. What are the barriers to prevent this? And we recently just authored a paper, which is kind of a state of the union on pharmacogenomics that highlights kind of 5 facets of things that need to actually be at a level of maturity to reach a tipping point to actually start to move into standard of care. Obviously, we have to have good lab technologies. We have to have clinical utility that's been proven. Implementation models have to be there that are appropriate for the clinical context in which they're delivered. We also have to have proof points for the economic value. It's not enough to just say this is scientifically interesting or even that patients feel better at the end of the day. We have to show that this ultimately saves money or it will never move forward. And then finally, we also need user acceptance. And user in this case is not just the patients, it's also the providers. So we did a little bit of a look at the market and sort of the history of where we've been in these different facets leading toward a tipping point for the broad-scale implementation, going all the way back to 2005. Where were we in these different aspects of pharmacogenomic implementation? And the progress has really been staggering when you think about where we've come from and where we are now, and where we were even just 3 years ago. The advancements in clinical decision support tools, in economic case studies, have really made it now such that it is not only viable, but it's advisable for large-scale implementation of pharmacogenomics. I'm going to talk mostly today, obviously, in the short time I've got, about clinical decision support tools, of which my company does play in that space. So I'm going to take the coward's route and show a video rather than a live demo. I was going to do a live demo, but I think it's probably best for all parties if I just hit play. But I want to highlight some economic outcomes that we've seen from some of the programs that we've run. We recently published in March of this year one of the largest economic studies in pharmacogenomic enabled comprehensive medication management that's ever been done. We've got 4 more in the pipeline, and I'll highlight some of the results of those today. I've heard this a couple of times. I think Carl actually used the term, it's one piece of the puzzle. I think we think about this in a very similar way. Pharmacogenomics itself is not sufficient. It's one piece of insight that can be gleaned in the overall care of medication management for an individual, but we have to look at all of those other things. I think there is a modality in which the delivery of just the pharmacogenomic results is valuable, but you have to think about that almost like an allergy, right? If I have a genetic allergy to a particular drug because of a variation in my genome, which I do, right? I don't take Plavix today, but I'm an ultra-rapid metabolizer on CYP2C19. I should never take that drug, and I should think about that, and so should my caregivers, almost like I have an allergy to that drug. It's a genetic allergy. That's useful, right? It's useful in the context of how I get my care today, if it's delivered at the right time. And I think Huda did a great job of showing exactly how that can be done within the context of actually prescribing medications. If we surface that information and say, don't prescribe Plavix to Scott, he's an ultra-rapid metabolizer on this drug or on this gene, here's why this is a bad drug for him, that's useful. However, what we really have done is to look at polypharmacy as a disease condition. Managing multiple medications for the same individual is a highly complex problem. And what Munir kicked us off with this morning, the eyes that are required in the ultimate system for decision support here, intelligence was that kind of top one, and we're just not there yet, right? We just don't have systems that are smart enough to be embedded in the EHR such that doctors don't have to actually think about what it is they're prescribing based on this additional evidence that we're giving them. However, there is a modality that can be employed whereby we leverage pharmacists in a way that we would think of radiologists or pathologists and empower them to be the case managers for those polypharmac, highly complex patients. So if we think about, again, those two modalities, we have this idea that let's put PGX information into the EHR, let's have that at the time of prescribing, so that when that prescription comes through and there's a genetic allergy, we highlight that. However, that's really not terribly sufficient, and quite frankly, physicians don't have the time to go play with a modeling tool. What we really need to think about is, in those complex cases, how can we enable a subject matter expert, and we advocate for pharmacists being those subject matter experts, to use clinical decision support. Now, why this is important, we don't think this is the way it should always be, but in order to get intelligent systems, we have to generate enough data. We have to generate data that can actually teach our intelligent systems what to do in the case of a future event, a future patient that looks like people we've seen before. And I think Coriell alluded to that as well. That data just doesn't exist yet at scale where we can actually train intelligent systems. So this is the baby steps into this world, but ultimately I do believe they will converge. So at Coriell Life Sciences, we've built an infrastructure that is very modular, and we service lots of different modalities. We have a pharmacogenomics knowledge base that's built up of industry consortiums like CPIC and PharmVar and all of those. We also layer on top of that an evidence curation team that's kind of pulling together the best of available evidence, so that we really have this database that's been battle tested, that's utilized by about 130 laboratories around the world today. So when we see human variation that occurs in nature that hasn't actually been well studied, that's now something our research teams have to go back and fix the database for. So it's been this heuristic process of constantly improving that database so that it is actually serviceable in wide-scale use. Combined with a drug knowledge base, which we can kind of plug and unplug, so companies like First Data Bank, Reed Elsevier, provide that basic information around drug interactions, anticholinergic burden, all the other aspects of drug information, and more importantly, risk that's important in the overall assessment of a given patient's plan. And then, of course, we need lots of information about the patient themselves. All of that flows through a risk assessment engine. We happen to have adopted a quantified methodology for actually assessing risk, and I'll show you how that shows up. It'll become very apparent when I hit play on that demo. But the outputs can be lots of different potential places. So laboratories and EHRs, payers that are interested in, how is this kind of a program performing, but also through the use of clinical decision support tools. The term agnostic was used quite a bit. We're agnostic of those endpoints, right? We really don't care where the data goes so long it is cared for in the right way when it gets there. But I'll show you one example of a clinical decision support tool that we've created. So at the end of the day, what we really have come to learn over the last 10 years of doing this is that it's fairly useless to give a physician anything other than the answer. And I apologize to any MDs that are in the room. And what I mean by that is it's not that there's not an ability to consume it, it's that there's not enough time to consume anything other than a direct recommendation. So what we have found to be quite valuable is enabling pharmacist teams to act as specialists, case managers, on behalf of a pharmacist who has a complex medicated patient. Those pharmacists use our clinical decision support tool, and the output of that process is a medication action plan. It is not a 54-page genetic report. It is usually a 2 to 3-page recommendation from a pharmacist who has gone through a process of comprehensive review looked at genetic and non-genetic factors, and has come to some conclusion about what pill should come off of the plan for that patient and what specific pill should be added instead. So that has been incredibly useful in systems where in the US we're dealing with a very fragmented system. Many of our customers they're not under a single healthcare system, and so the output has to operate at the lowest common denominator level, which is really get an answer to the physician that's actually caring for this patient. I'm going to take just a few minutes. I'm going to show you what our clinical decision support tool looks like. Hopefully this doesn't sound much like a sales pitch. That's not the intention today. This is really, here's where our experience has led us to create a modality that has proven successful from an economic standpoint, and then I'm going to jump right into some of the economic case studies. So if I go forward, hey, there we go. And like Carl, I'm going to sit down so I'm not staring at you.
GeneDose Live is a clinical decision support tool built specifically for pharmacists and physicians. It supports comprehensive medication management reviews, including the use of genetic test results. GeneDose Live can be accessed with any internet-connected computer or mobile device. Let's have a brief look at how a pharmacist might use the tool to manage the medication plan for a patient. After logging into the system, the pharmacist is presented with a list of all of their patients. Each patient record lists some of their key risk scores and an overall status for that review. Let's have a look at Gary Herbert. Gary is a 78-year-old cardiac patient who is medicated for a series of related conditions. Last year, Gary had a stroke and has been having difficulties with his medication plan. Gary recently took a pharmacogenomic test that provided guidance on hundreds of drugs. Gary doesn't take all of these pills today, but because our DNA doesn't change, this information can be used to steer his care for the rest of his life. Along with the DNA results, we can identify other potential risks for Gary based on his age, his lifestyle habits and diet, and the medical conditions he suffers. We can then input Gary's current medication plan, including any non-prescription vitamins and supplements he may be taking. A wealth of information is available to the pharmacist for each drug, including reported side effects, indications, contraindications, precautions, market pricing, and even any product education sheets that may be important to provide directly to the patient. Once we have all of Gary's information, GeneDose Live will then analyze the plan for any risks that might be important for Gary. Safety and efficacy concerns are displayed graphically with each type of risk represented in a different color. Genetics in blue, drug interactions in yellow, and so forth. The size of each bar indicates the severity of the concern. In Gary's case, his DNA makes him a poor metabolizer for Plavix and will render that drug ineffective for him. The FDA includes this pharmacogenomic concern right on the drug label. Similarly, Gary's tests also show he is an ultra-rapid metabolizer for codeine, which will significantly increase his risk of adverse reactions. Other medications in his plan also have issues unrelated to his genetics. Each of those risks is further detailed below, categorized by their degree of concern. Also available here is a roll-up of the frequencies of side effects reported to the FDA for each of the drugs in Gary's plan. When Gary mentioned he wasn't very good at taking his drugs all of the time because they make him dizzy, we can use this tool to help identify the likely culprit. Once we've identified drugs that may not be well suited for Gary, we can then use the alternatives modeler to find medications that are more likely to be safe and effective. Since we know that Plavix won't work well, we can ask GeneDose Live to show us all of the alternative medications for that therapeutic intent. In this case, we'll try ticagrelor as a replacement since it does not present any genetic concerns for Gary. Selecting the new medication and updating the alternatives now shows us what impact that change has had on Gary's overall plan. We've eliminated the genetic risk for his blood thinner entirely. His prescription for Lexapro looks to have some significant drug interaction issues, which should also be addressed. In this way, we can continue to work his plan, looking for choices that will have fewer safety concerns. Once complete, we can save the new regimen and alert Gary's physician to the new recommendations using a medication action plan. By selecting the starting regimen and the new suggested regimen, GeneDose Live will present a worksheet allowing the pharmacist to provide some details to the recommendation. Each change can be tagged with a reason for the change, as well as any specific remarks that may be important for the prescriber. Any general notes can also be added here before finally generating an overall plan document.
So the output of that system is a number of different types of reports. The first that was only briefly up on screen was the medication action plan, which is intended for what is effectively an uninitiated physician. In most of our programs, the patient is actually initiating this process, and so their direct caregiver, their GP, is actually the recipient of a process that they didn't even know was occurring. The other type of report, which is quite usual in these circumstances, is what we call a medication compatibility report, which is intended for the patient themselves so that they have an understanding of what drugs they should avoid in the future based on the pace at which those drugs may or may not move through their body. It is really intended to be non-prescriptive, so they don't go out and try new pills on their own, stop taking pills on their own without actually consulting their physician, but it empowers them to have better conversations with their doctor in the future. All right, let me talk a little bit about some economic outcomes. So these programs, you'll notice that this whole model is not geared toward implementation in hospital systems. Right? Pieces of this certainly can be, but in most cases we've been implementing as a stand-aside to wellness benefits within self-insured employers, pension funds, government agencies, groups that have to care for large populations of individuals, where we do population health analytics work to target who should be eligible, who shouldn't be eligible, things like that. But just a little on some of the economic outcomes. We've kind of coined this term of MA3, which is how do we actually measure the value of these programs? We have to look at the cost that occurs when a patient doesn't take the pills they've been prescribed. So issues that would be preventing medication adherence, medication appropriateness, so did we actually choose the pills that are going to actually be safe and effective for them, and then medication adverse drug reactions. So obviously we want to avoid those safety concerns. The value of getting the meds right is not generally borne out in the cost of the pills. We do see an overall reduction in pill cost throughout the course of programs like this, but it's not because we put people on cheaper pills. In most cases, it's because we've reduced duplicative therapies, that this concerted look at someone's medication plan has surfaced that you don't need to be on this pill because you're already on another one that your other specialist prescribed. Or when we get you on the right statin, then we can take you off of a pill that was prescribed to counteract the side effect of you being on the wrong statin in the first place. So what we're really looking for in these programs is if we are doing a pure apples-to-apples comparison where we do a cohort comparison of propensity-matched individuals, where the only thing different is their participation in this overall comprehensive medication management empowered by PGX program, how much of a difference do we see? So back in March of this year, we published a paper looking at one of our longer-running programs, which was started back in 2017, which was the Kentucky Retirement Systems for Teachers in the state of Kentucky. And there they have about 35,000 retired teachers. About 12,000 have actually enrolled in the program, so really nice uptake there. And at the time of this analysis, which was after 32 months of intervention availability, about 5,200 individuals had enough data pre-program and post-program to actually be part of the analysis. Just a little bit more on them. So at the time of my slides here, 10,000 individuals. We're up to about 12,000 that have enrolled. And this was, again, a retrospective review of medical claims for those individuals with both 12 months pre and at least 12 months post. So 5,200 that were compared to 22,000 controls. Very similar intervention and control groups. We did both a propensity score matching as well as matching on things like Charleston Comorbidity Index, basic demographics, things like that. And the N was obviously big enough that it was highly statistically significant. The results were really nice. We got to see a visual bending of the cost curve. If you look at the intervention group in green, the control group in the kind of gray there, the intervention group was more expensive to begin with, but over the course of that 32-month period, they became cheaper. And they predictably so, which was quite nice. But the large majority of that cost reduction came from hospitalization reduction. So it was emergency department visits, inpatient and outpatients that really drove the reduction in overall cost. For the Teachers Retirement Systems of Kentucky, that equated to $37 million over the course of the 32-month period. That was an ROI for them of better than 10x, which was really quite nice. So that's been published, that's out there, peer-reviewed, it's in the Journal of Personalized Medicine. Wanted to also highlight a couple of others that are in the works now, and some of these I just can't mention names until we actually get the publications in for peer review. But one is an international hotel chain, a name of which you will all certainly know. We did an initial study of 500 employees there. So this was employees that were identified to be polypharmac, but otherwise there were no criteria for their participation. This was direct outreach to those employees through email and mailers to their home to encourage enrollment. After 12 months, we saw a 6.1x ROI, which is quite nice for them. We had sustainability of the program economically after 10 months. Some very similar results in a large manufacturing company, 1,000 employees in that one, 10.1x after 18 months. Quest Diagnostics, since we've already published 2 papers with them, and the third is in process, that one I can name, and that is for their own employees. They achieved a 6.4x ROI after 12 months. And then also the state of New Hampshire, which is we are running a program for all of their current state employees and all of their retirees, which is about 55,000 eligible members. That's another one where we've got better than 2,000 enrolled folks now, and we've just gotten the data feeds in from their third-party administrators. So all of those will be pending publications that we expect to have out either by the end of this year or first quarter next year. So really good stuff. All right, let me pause there, see if we have any questions.
Any questions? So maybe I can start, or go on. Yes, yeah. No, you start.
Lots of people talk today about getting more and more sources of information. So about drugs, about kidney function, about liver function, whatever. And obviously one of the problems, as you add the number of variables, is the number of possible interactions go up. And you're never going to be able to model those interactions because you will never have enough people with that particular set of drugs and those particular set of disorders. And even if you could, the modeling just gets five-way interactions just are impossible. I just wondered, how do you think about that? I'm just sort of interested.
You've really nailed it. And back to my original point about the systems just aren't intelligent enough today, and we don't have enough data to actually train systems to actually look at all those variables. We've had to scope back some of those things. Like in most of our programs, we don't have creatinine clearance data. We don't have renal function data. What we are really focused on is what can we provide over and above what they're getting in standard of care today that is a tractable amount of variables. And so things like, their age, their conditions that they're suffering, anticholinergic burden, the other drugs they're taking, which once you start looking at the other drugs, that brings in, 6 more variables on potential interactions and, competitive inhibition between drugs. All of that, we've done just shy of 2 million pharmacogenomic tests. We've done this program now on a couple 100,000 people. We're now getting to the point where we have statistical significance on actually tracing those uncommon connections, but we're letting the data drive it rather than us, applying what we think should be the case, because what we've got is genetic information all the way through outcomes. So when we see someone with this particular mutation, what worked to actually provide, patient improvement and economic benefit. So, but I think you do have to scope it, right? I think you're exactly right.
For a really good presentation. I had a question about the GeneDos Live user interface. Thank you for sharing that. How does the user enter the additional data points such as age and other medication or comorbidities? Are they pre-populated through integration with their EHRs, or are they manually populated by the pharmacist that's using it?
Populated. So in every one of our programs, we have some connectivity to either an EHR or an eligibility file or claims data or wherever it can be. And in some cases, it's self-reported during the enrollment process. So there generally isn't a source out there for lifestyle information. You're not going to find, do you take grapefruit juice with your morning tea, as something that sits in the EHR. But it's important for this. So generally speaking, it's this combination of different sources that come together. Yeah, but we generally don't have pharmacists keying anything in. Yeah.
Did you have a question, John?
Very much for that. It was really nice to see your health economic data. I think that's a piece of the puzzle for pharmacogenetic implementation that's been missing. So it's really, really nice to see that. I had a question just about the video. You demonstrated a risk graph that went from 0 to 100. Could you explain what that metric actually is?
Yeah, so it should not be seen as a giant echo chamber. It should not be seen as a 0 to 100 scale. That's not a percentage basis. That's a cumulative score of risk based on the number of factors that fall into those severity buckets. So if you have a genetic severe concern, you're going to get a quantitative score of 20. We assumed that that lexicon for risk based on the underlying drug database that we use today that has a very similar scale. So what we did was we went back and scored the pharmacogenomic concerns to those same severity risks and applied that quantitative score there. It's not intended to say if you are a 70, you're about to die. What it is to say, when a pharmacist looks at it, they have a graphical representation that makes the really important bits jump out at them. So that's the intent of that.
Hi, Scott.
Hey, Huda.
Thanks for the presentation. For a pharmacist, what is, based on your experience, the average length of time of this comprehensive medication session? And does it require the patient to be there? You mentioned the grapefruit juice story, to know all of the other variables that might not be in the EMR.
So we track all of that. So we're right now 23 minutes is the average case review. That usually includes a phone call with the patient. So the things that generally still have to be reviewed direct with patient are vitamins and supplements that aren't going to actually appear in the EHR, lifestyle factors, previous medication failures. That's generally something that's quite useful to know during that conversation. So there's a whole separate system where we do online booking with pharmacists. We have about 300 pharmacists that are on the bench with us at any given time that do those reviews. Yeah, sure.
So some of the data they used to input for the pharmacogenomic guidance included data from PharmGKB, PharmVar, and CPIC and Dutch guidelines. So do you use all of that data, or do you limit it just to CPIC and the Dutch pharmacogenomic guidelines? Because obviously some of the data in the PharmGKB is of different levels.
That's right. And some of it is actually contradictory, unfortunately. So we've published on a process by which we do evidence curation and what we call chain of evidence. And if you want to put yourself to sleep and you Google Coriell and Phasis, Phaesis, you will get a paper that details all of that process for evidence curation and how we come to which one do we actually use in the clinical product.
Okay.
Yeah.
Yet you don't use creatinine clearance, you said.
But we don't, only because of exactly the reason that we talked about before. It's a variable that, one, we don't often have in this process because we generally are operating outside of the
You don't have the
EHR.
lab data.
We typically only have the pharmacogenomic data.
Okay,
Yeah.
okay, because given that literature shows that in 50% of cases, clinicians ignore the renal function
Yeah.
and still prescribe drugs they shouldn't prescribe.
Yeah.
So,
There are no perfect systems out there today. What we're really trying to do is let's take the next step so that we can build the more intelligent systems down the road.
okay, well, thank you very much.
Yeah, my pleasure.